Okay, hello everybody. As I'm sure you're well aware, Ray and I are not in class. We're both chilling like villains at SFN on the East Coast, so we decided it'd be good to work together as a group of two, and that way we can both just kind of pre-record and share that way. Okay, and the paper we chose to review is a transcriptomic and epigenomic cell atlas of the mouse primary motor cortex, highlighted here. Okay, and let's just jump in. We'll start with the introduction material. So there are now many ways to quantify cell types. These authors noted this and decided to combine 9 data sets collected from um, mice primary motor cortex into one atlas. The 9 data sets involved different techniques with different strengths and weaknesses. These techniques included single cell RNA-seq, single nucleus RNA-seq, single nuke DNA methylation, and single nuke ATAC sequencing. They ran these combined data sets through multiple clustering algorithms and did cross-validation to further demonstrate the robustness of their cell type classifications. This combined atlas resulted in over 600,000 data points, which were reliably clustered into 56 cell types. Okay, and I said they had different strengths and weaknesses. You can kind of see these kind of get you into 2 broad categories given these 9 seeks methods. You get a lot of cells down over here, but not as many fragments per cell. And you kind of have this opposite problem in strength in these techniques, where you can get a lot of fragments per cell, but obviously, right, this comes with the trade-off of less breadth. Okay, so the authors thought, let's combine both of these and see what we get. Okay, and so first off, there were 9 data sets, but they just wanted to look at the transcriptomic first, so they made that's 7. These first 7 listed here, and makes the transcriptomic atlas. And when they combine these 7, they get a total of 116 cell types, 90 of which are represented in all the data sets. So some of them had holes in some data sets. And of these 116 cell types, roughly 90% of them that were identified, they were found to exist in independently made data sets from single cell RNA sequences. These independent data sets were of the anterolateral motor cortex and the primary visual cortex. So this is a nice sanity check that things are going well. There's high overlap with other cortical regions, one of which was it's nearby. Oh, and before I go on to the next slide, we're looking at UMAP projections here in the middle and on the right. UMAP is just a dimensionality reduction technique, very common in machine learning nowadays. The real data space is higher dimensional than just the 2 we can see here, but we can still kind of get some interesting data from this projection that we're looking at. One of which is in the middle here. We're coloring by the 1 through 9 shown on the left. So it's coloring by sequence type. And what this shows is that these clusters are obviously multimodal, since these different sequence data set types are commingled. And on the right, we have the exact same projection. We're just instead looking at everything labeled by actual cell types. Okay, let's carry on. So we're still looking at the transcriptomic atlas, the first 7 data sets combined. They found total of 70 cell clusters, given the roughly 120 cell types. And they decided this based on how robustly these same clusters were found when using a different cluster resolution. So there's this, the replicability of clusters increases as you use fewer clusters, but you don't want just very few clusters. We know there's going to be more than just 5 clusters. So this leads to a tough decision of how many clusters do we include. So one way is, right, you can just look at reproducibility. You can run through a bunch of different cluster resolutions, allow only so many different clusters to be identified, and see across all different configurations how reliably the clusters are found. And so there's a trade-off, like I just outlined. If you have a bunch of clusters found, you tend to have low reproducibility. So that's one part of how they ended up with 70 cell clusters, but they also use what's called cross-validation, another common machine learning technique. And they did this to further demonstrate the robustness of these clustering techniques. Um, this isn't really the focus of this class, these kind of computational methods, but I think I can convey uh, what we're looking at here. So we've got error of the clustering techniques. So the higher you are, the worse you are. And so we're kind of watching learning happening in a sense, kind of here. So if you let the number of clusters learned be low, you end up with higher error. That makes sense. And this is just while training. So we let it look at a bunch of the data, and wow, the training data is really good if you have 1,000 clusters. But the problem is, this testing. This red line is showing you kind of a generalizability of your what you learned during training, because you take some of your this testing is from data that the algorithm never saw, but comes from the same underlying distribution, right? These testing data set comes from our seq data, but we just never let it be trained on. So when these two diverge, you kind of think, well, I'm probably overfitting to my training data, and I'm learning a bunch of spurious correlations that don't actually exist in the broader context. And so you see these two kind of diverge around 70. So this is more evidence for using 70 when also combining it with this data here on the left. Okay, and they use this transcriptomic data to question the common dogma of whether or not layer 4 exists in the primary motor cortex. But they pointed out that there's mixed findings when considering recent anatomical studies, and this just adds to that kind of fire, I guess, because they found 2 IT clusters with a total of 100,000 cells co-expressing markers, traditional markers for layer 4 and layer 5. So that's going to be CUS2, RSPO1, and RORB for layer 4, and FESF2 for layer 5. And so all of these except for let's see, CUS2 are down here, and you see that these two especially have high expression, and layer 5 even a little bit less. But they ended up calling this layer 4/5 IT, and this actually ended up representing 20% of all excitatory neurons in the data. So that's pretty interesting. Okay, so now they shifted gears, in those 2 other data sets that we haven't talked about yet were epigenomic data sets, and they combined these into one epigenomic atlas. And we'll combine this back with the other 7 to make the grand atlas here in just a second. But just focusing on the epigenomic stuff, they found that the differences in methylated regions between all cell types accounted for 8.3% of the total genome. And among these different cell types, 28 to 89% of accessible DNA regions overlapped hypomethylated regions. And so you can see that here. We've got accessibility as the whole shebang. And then the blue is the hypomethylated regions. And so I guess this layer 2-3 IT is going to be our extreme of overlap between accessibility and hypomethylated regions. So these fit the traditional, easy to understand view that, if the DNA is euchromatic and open, that it's easy for transcription factors to get in. This SST is kind of the opposite, and it represents the lower bound here, that's 28%. Okay, and they also looked at MCG, the DNA methylation, and the open chromatin is reported by ATAC, and they found a bunch of enhancer regions, about 250,000 of them. And they were very often over 2 kilobases away from the nearest transcription start site. So I found that super interesting. You have to consider consequences far away linearly when in 3-dimensional space. Okay, and like I alluded to, they ended up making the final atlas by combining the transcriptomic atlas of 7 data sets with the 2 epigenomic data sets to make one big granddaddy atlas. And so they used 2 methods to do this. They used Liger and single cell fusion to create a unified multimodal cell census. And then they compared how similar their findings were. They both identified 56 cell types, and these results agreed with the aforementioned analyses as well. During this discovery, they could actually get about 600 clusters, and still get good generalization as measured by that cross-validation I showed earlier, where those test and training diverge. But they figured this was actually capturing natural continuous variance within single cell types, kind of over-splitting things. Even though they were unique, they thought that this was just, yet again, spurious correlations. There was statistical power in there that shouldn't be differentiated. And so what this confusion matrix on the left is showing for each cell type how much they agreed. So this is a confusion matrix, common in machine learning again. A perfect agreement would actually have all of the heat along the diagonal here. And so these are, the main takeaway is there is some disagreement about what Liger spits out in the 56 cell types and what single cell fusion spits out. But really, I think the takeaway is that they largely agree. So that's really interesting. And then in conclusion, these authors created a primary motor cortex atlas that combined the strengths of different broad and deep sequencing data types. They did a bunch of subset comparisons to support the notion that these data sets can be appropriately combined and clustered. And they identified more than 50 fine-grained neuron types by correlating mRNA transcripts, gene body methylation, and accessibility peaks. This work overall represents a two-pronged approach, which combines broad sampling of diverse cell types with deep sequencing to precisely characterize gene expression profiles for each cell type. And the authors encourage us, anybody doing this stuff, to go forward to use this technique as well. And that's it for us. Thanks, guys.